This week, we are going to talk more about proportions beyond the binomial test. If you recall, the binomial test, seen here, gives us an exact p-value for a comparison between our observed probabilities and some underlying null probability. We can go further by testing whether a particular set of observations is from an expected distribution with specified parameters. In other words, how well does our data fit to our hypothesized distribution? To do this, we employ the chi-square test, which is the sum of the squared differences between the observed values and the expected values, divided by the expected values. We can take a simple example from population genetics. Mendel developed the principle of independent assortment, which states that alleles will segregate independently into gametes in a dihybrid cross, with a phenotypic ratio of 9:3 to 3:1. For example, see this dihybrid cross table of big B little b and big E little e in hybrid flies. The B gene codes for body color and the E gene codes for eye color. The dominant body color is brown and the recessive color is black. The dominant eye color is red and the recessive eye color is brown. See that there is a 9:3 to 3:1 ratio of the possible phenotypes expressed by offspring. However, we now know that sometimes certain alleles are associated with each other, or in other words, are not independent. We can test to see if alleles are linked using the chi-square goodness of fit test. In this example, we take flies that are heterozygous for both the B and E genes. We then perform 100 dihybrid crosses and get the following offspring phenotypes: 50 brown-red, 20 brown-brown, 20 black-red, and 10 black-brown, for a total of 100 individuals. We can use the chi-square test to see if these genes independently assort. If we expect a 9:3 to 3:1 ratio, we can calculate the expected phenotypes of the offspring. We would expect around 56.25 brown-red offspring, 18.75 brown-brown offspring, 18.75 black-red offspring, and 6.25 black-brown offspring. Notice these numbers also add up to 100. We can then calculate the chi-square statistic using the equation. Here, chi-squared equals 50 minus 56.25 squared divided by 56.25 plus 20 minus 18.75 squared divided by 18.75 plus 20 minus 18.75 squared divided by 18.75 plus 10 minus 6.25 squared divided by 6.25. And we end up with a chi-squared of 3.11. Of course, this is just a test statistic. We now need to calculate the p-value. Unlike the binomial test, which gives us an exact p-value, the p-value for the chi-square test is approximated using the chi-square distribution. We can calculate the p-value by using the p-chi-square function, or PCHISQ function, in R. We need to add the chi-square value of 3.11, our degrees of freedom, which is the number of categories minus 1, in this case 4 minus 1, or a degrees of freedom of 3. And the chi-square test is a one-tail test. We need to set lower.tail equals false. If we run it, it gives us a p-value of 0.375, which is much larger than 0.05. Therefore, we do not have evidence to say that our sample is different than the 9:3 to 3:1 ratio. Therefore, these alleles do independently assort.